So controlling neurons has been done for a long time. People use electricity to stimulate neurons. People use pharmacology to modulate neurons with drugs. But drugs are slow, and electricity goes everywhere. So how could you activate or shut down just one neuron? If you could activate or shut down neurons, then you could really figure out how they contribute to their circuits, right? You could turn on some cells and figure out what they can initiate. You could shut down cells and figure out what they are needed for. And so light can be aimed, unlike electricity. And light is a lot faster than, well, everything else, actually. So it has enough speed that we can keep up with brain activity. And you can also put probes into the brain. We talked about electrodes earlier, but you could put optical fibers into the brain, too. The brain doesn't feel pain. And the hard part, in a way, is knowing how to make neurons sensitive to light. So it turns out, all over the tree of life, you can find organisms that have molecules that convert light into electrical signals. If you've ever looked out into a salty water and seen sort of an orangish color, that's because of microbes that have a pigment that absorbs light, and when hit by light, moves a charged particle, a proton, from one side of the cell membrane to the other. And so it's a way of storing energy. There are other molecules that pump negatively charged ions, like chloride. And then finally, other molecules that also sit in membranes that let positively charged ions into the membrane. So these are all proteins. They're encoded for by small genes. We can put those genes into the genome or into the cell using gene therapy vectors. And over the years, what we and our colleagues and collaborators have found is that some of these molecules work really well. Some of the light-driven proton pumps can pump protons out of neurons. And when you do that, you silence the neurons. Light-driven chloride pumps pump chloride into neurons, also shutting them down. And the light-driven ion channels let sodium, potassium, calcium, and protons into the cell. And that can activate the neuron. So there's all sorts of things you can do with these molecules. There's a disease called narcolepsy, where people fall asleep at random and inopportune times. And in these patients, a small cluster of cells has died off, cells that make the peptide hypocretin or orexin. And so the Yamanaka group made a transgenic mouse that has a light-driven chloride pump in just these hypocretin or orexin neurons. They then aimed an optical fiber at these cells, connected to a laser. And when you turn the light on, this is the probability of being awake. The mice start out awake, and then they turn the laser on around here. And as you can see, the mice all completely fall asleep. And then the light turns off, and bam, they all wake back up. So you can really use this to prove that a specific cell is involved with a specific behaviorally relevant condition. So what we've been doing recently is figuring out how to make this even more powerful. Red light can go very deep into the brain, right? Because red light is not absorbed by blood. And so Amy Chuang, who recently finished her PhD in our group, worked on a molecule that was more red light sensitive. We named it JAWS. It's a long story. We find these molecules by looking at genomes and then mutating the things we find. But JAWS, because it's red light sensitive, allows us to non-invasively shut down neurons. We can shine light from an optical fiber across the skull of an awake behaving mouse. And as you can see here, this is percent suppression plotted versus depth. We can shut down neurons many millimeters deep in the brain. What about activators? So this is some work from the Fiorillo group. What they were doing is studying dopamine neurons. I think we've all heard of dopamine neurons. They're involved with reward, addiction, learning, all sorts of stuff. And also involved in their absence with Parkinson's disease and other pathological conditions. So what they did was to take a transgenic mouse and virally deliver the activating opsin, known as channelrhodopsin 2, to the neurons. And then they would put a mouse in a box, and every time the mouse goes to this spot, it gets a pulse of light. And every time the mouse goes to this spot, nothing happens. And so you can see what happens. The mouse goes, gets a pulse of light, pokes his nose, gets a pulse of light, pokes his nose again, gets a pulse of light. And so the mouse is basically working for light. And so that allows you to prove that activating these cells is enough to reinforce the behavior and make the brain do more of what it was just doing, at least in the context of this behavior task. So recently, on the tools side, we've developed a red, in fact, nearly infrared activator of neurons that we call Crimson. This is, down here are action potentials from the mouse visual cortex. And you can fire spikes with 735 nanometer light, which is quite far to the red. We also solved a problem, which is, can you make a very fast molecule that's also very light sensitive, a kind of good all-around option for activating neurons. And so we call this one Chronos. And interestingly, Chronos and Crimson, these two molecules that we just talked about, if you use them together, you can do two-color control. You can put Chronos in one pathway and activate it with blue light, Crimson in another pathway and activate it with red light, and see how two sets of neurons work together. And the actual reason why this works is a little bit complicated to go through. But this is work that Nathan Klepetke, who also recently graduated from our group, did. In intact brain circuitry, you can activate two separate populations with blue and red light. All of these tools you can again find on our website, syntheticneurobiology.org. We've developed a whole pile of other tools to make these tools easier to use, including wireless devices, ways of using optogenetics in the conjunction with functional MRI. Some of our collaborators have developed transgenic animals. And then finally, devices for 3D light delivery, so we can be more naturalistic. And just to end on a note that this is all a really interdisciplinary, if not omnidisciplinary endeavor. Really working from all these different angles, physics and nanofabrication and optics and chemistry and protein engineering and genomics and so forth, trying to build these tools. But the working model that we have in our group is, let's assume that sometime, maybe 10 years, maybe 20 years, maybe more from now, but not longer than 40 years, the brain is completely solved. How do we get there? Thank you. Great question. Yeah. How do we get one kind of cell sensitive to light? Let's pick this example here. So what does this mean? There's a molecule called Cree, and Cree will bind to certain sequences in DNA and delete or otherwise alter the stuff in between the Cree target sites. And there are hundreds and hundreds of transgenic mice that express Cree in different cell types. Basically, the way this works is suppose here's the cell that makes dopamine. It has a gene called DAT, which stands for dopamine transporter. And in the genome, that gene is regulated by a little regulatory sequence called a promoter. And some factor, we don't know what it is, factor X let's call it, binds to that promoter and turns on the DAT gene, and now this becomes a dopamine neuron. So what we do is we take that chunk of DNA that's the promoter. Usually people just take some number of bases of DNA in front of the gene and hope that it works. And then you could put Cree after it and then put this back into the genome. It just goes into some random part of the genome for the most part. So now what happens? This is in this cell, this is in this cell. We have the promoter and Cree, and here we have the promoter and Cree. Well, here in this cell, there's no factor X. We know that because if there was factor X, it would have turned on dopamine transporter. So no factor X, no promoter activity, Cree is off. This cell, though, has factor X. So it binds to this promoter because it's the same as that promoter, right? So Cree is on. That's part one. You can have Cree on in cells you like. Basically by taking the piece of DNA in front of the gene that's on in those cells, pasting it in front of Cree, putting it back in. Okay. Now, what about part two? How do we actually get the opposite in there? I'm going to draw a version of this that's easier to explain, but is not exactly what is here. Cree binds to a sequence called LOX. So Suppose you have a promoter, and this is a generic promoter, just some boring old promoter that drives gene expression. And then you have a stop sequence here. There are various stop sequences that you can get, that you find. You can put them in. And here's the opsin. Let's see, it's channel rhodopsin 2, which is the one that was used for activating the dopamine neurons. Cree will bind to the locks and delete what's in between. So in all the cells that don't have Cree, like this cell down here, no Cree, the stop stays. And so something will try to make this gene, but it'll hit the stop sequence and stop. However, in the cells with Cree deletes the lock, stop, locks cassette, and now this promoter can drive the expression of the opsin, and now the cell is light activated. So 2 parts. This is a very common strategy right now in all of biology. You have what's called a driver, which expresses Cree in a certain cell type, and a reporter, which is a little bit of a misleading concept right here, because this is not just reporting, it also lets you control, right? But they call it reporters. And so by combining the driver and the reporter, you can then express any gene you want in any cell that has a well-known existing gene. So nobody's used this in people because it's a gene therapy. There are no gene therapies approved in the US. There are people trying to see whether it could be a therapy for patients with blindness, for example. Neurotechnologies have been around for a while and haven't always been used properly, you could argue. Over 1/3 of a million people have had some kind of neural implant for stimulating their neurons so far. And then, of course, millions of people take pharmaceuticals that alter brain activity as well. I think you're asking 2 questions. One is, could this be used in people? And the other is, could that go wrong, right? So the answer to the first is, for certain forms of blindness, there are 3 companies that are trying to see whether you can actually treat blindness with these molecules. People lost their ability to see, could you install these molecules and help them see again? As far as the should people use it aspect, that's a question that has to be asked for any biotechnology, I would argue. People can edit the genome now, people can make stem cells from almost any person. All of these technologies have to be thought about, openly discussed, and ethically used. The brain is really complicated, right? So if you look at this picture, they turned off the neurons and the mice fell asleep. They turned off the light and the mice woke back up. But they're still a bit groggy, right? They go to sleep again. So there's very complicated time dynamics, right? All the cells in the brain are interacting in a very complex way. And people have shown that if you stimulate even one cell in the brain, you can change the entire brain state. So it's a very complicated set of questions. The original activator that people used, if you recall, was using blue light, right? So in this movie, the animal pokes his nose in and gets a pulse of blue light. The reason that JAWS and Crimson are important is because they can be activated with red light. And red light can go much deeper in the body than blue light, right? If you take a bright light and shine it through your finger or something, it looks red, right? We don't look blue. The red light goes through. So you can perturb much bigger volumes of brain tissue with red light than with blue light.
And is crimson just like a different red than JAWS?
Crimson is a molecule that we found through a massive genomic screen of looking at over 1,000 plants, and it's spectrally shifted. So this is current on the y-axis and color on the x-axis, and this is the crimson response curve. It's shifted to the red. Were you asking how crimson is different from JAWS? So JAWS is a silencer, right? It pumps negative charge into the cell. Crimson is an activator. It brings positive charge into the cell. So you need both. JAWS and crimson have nearly identical spectra, but if you wanted to do 2-color perturbation, the way to do that would be to use Chronos to activate neurons with blue light, and crimson to activate with red light, or JAWS to silence with red light. Yeah, I think that would work. There are groups already trying such experiments in vivo. Suppose that X years from now, the mouse brain is solved, right? We have a good map. We've recorded the whole thing. We can simulate it. Maybe that takes a couple decades. Who knows what, right? It's really hard to know how much will generalize to the human brain. Certainly some things will, right? Mice have emotions. They make decisions. They have memories. On the other hand, it's not clear, about language and higher order cognition and all sorts of stuff. So I think there's a couple strategies that could be brought to bear. One is, at MIT, we're starting to try to get the marmoset going. It's a really small, 1 kilogram not New World primate, not as smart as the Old World primates like macaques and so on. But it's really tiny. And so one idea is, could we adopt more model organisms into neuroscience that we can try to bridge those gaps? And that's important too, because like if you, even for therapeutics, right? If you develop a therapy, you only try it on a mouse, and then you go to humans, there's a very high failure rate, right? We can barely cure anything as a brain disorder goes. So I think that one strategy is to have more intermediate species. And it could be that the massive convergence in neuroscience over the last 15 years on mice is a temporary thing. We might eventually, now that it's so easy to put viruses into different animals, and this whole CRISPR business for editing the genome and so on, maybe what's going to happen is that the mouse convergence between 1995 and I guess now will expand back out, and suddenly people are going to start working on different species. There's a second point though, which is maybe we should try to develop better human neurotechnologies. So we have a project that's really early stage to try to develop neuroimaging strategies that could image without requiring neurosurgery and without damaging the brain of a human at all. Can we record neural activity with single cell, single spike resolution? And this is very early days. I can describe one of the ideas if there's time, but all right, here's the idea. Suppose we could put an optical fiber into the bloodstream. It will not enter the brain. It'll just go through the bloodstream and sit in the bloodstream. Suppose we send a pulse of light down the optical fiber. Now, suppose that every time a neuron fires, the optical fiber changes so it becomes a little bit more like a mirror and reflects light. So let's walk through that, and then we'll get to the implementation. Send a pulse of light. Time equals 1, time equals 2, time equals 3, time equals 4. Oops, a neuron fired. Reflection, 5, 6, 7, 8. So from the time of flight of light, you can figure out where the neuron was. Now the question is, how do you have a dynamic change that responds to a neural activity? To be honest, we're not exactly sure how to do this. So we wrote a manuscript and just posted it on the internet. And if anybody wants to join in and collaborate, the more the merrier. But here's our concept so far. This is a cross-section of the optical fiber. There's a thin insulator. The insulator is very thin, so that all the neurons' voltage is concentrated over a very thin area. You get a very steep electric field. Then we have our optical fiber, but it's actually made out of silicon. And when you have a very strong electric field next to a silicon piece, you get an accumulation of charge. And when you accumulate charge, you change the refractive index. And when you change the refractive index, it reflects light. So that's one concept that we're trying to get out there. But you can imagine then that you could, somebody would have an injection, these fibers would be staked into the brain. You could record the neural activity all along the fiber, because the time that the light takes to get there tells you which neuron it was. Now we just have to figure out if it's possible to make this. But at least we're trying. We're trying to get into the human neurotechnology changing. Yes?
Can we also find a way to turn on some cells?
As opposed to?
Turn it off.
Oh, yeah. Yeah, so that mouse that was working for light, its neurons are getting activated by light.
Okay.
Yeah, let me go back to that slide. So in this slide, the light-driven proton pumps pump protons out, and that shuts neurons down. The light-driven chloride pumps pump chloride in, that shuts neurons down. The light-driven ion channels let positive ions in, that turns neurons on. And that's the molecule that we used in the mouse working for light experiment. Yeah.
My second question is, can we use some of this technology for learning, for help in learning?
Oh yeah, I mean many groups, even at MIT, have used these molecules to study learning in mice. And yeah, there's a group here at MIT that they have a mice learn something, and they turn on the opsin only in cells that were used during that task. And then later they activate those cells with light, and they can cause the learning to transfer, they can cause the memory to be recalled, they can cause false memories. Yeah, yeah, that's been done. Yeah, literally hundreds of groups have used these tools to study the brain, and they've even found neurons that trigger memory recall, shut down epileptic seizures, all sorts of stuff. All right, well, thanks.